The overall goal of the following experiment is to deliver antisense oligonucleotide morpholinos or other development-perturbing molecules directly into the zebrafish otocyst at the 1-day or 24-hour post-fertilization stage. In this example, we use morpholinos to the earliest neurotrophin responsible for statoacoustic ganglion development, an immune system cytokine called macrophage migration inhibitory factor, or MIF, to determine if morpholinos to this molecule affect inner ear development or neurogenesis. This is achieved by first introducing morpholinos into the lumen of the otic vesicle using microinjection. Then, electroporation is performed, which facilitates the penetration of morpholinos into the cells of the otocyst from the lumen. Next, the embryos are raised at 28.5 degrees Celsius in order to observe the effects of the morpholinos on inner ear development and neurogenesis over the next few days. Results are obtained that show a decrease in inner ear sensory hair cells, as well as a decrease in innervation by the statoacoustic ganglion, based on immunohistochemistry and phalloidin staining, which label acetylated tubulin in kinocilia and the actin in stereocilia on the hair cell surface, respectively.
We originally developed this method of injection and electroporation of morpholinos in the chick embryo, and a very talented undergraduate student in the lab, Katie Holmes, successfully scaled it down to zebrafish dimensions. The present research team, including Shen, a postdoc, and Wyatt, a graduate student and a former postdoc in the lab, Deb Thompson, refined this technique for zebrafish. This method hopefully can answer key questions in the fields of developmental biology and auditory research, including